Our next talk is by Geert de Vreede from the University of California, Berkeley, and the title of his talk is the Drosophila tumor suppressor, TID Alg3, controls TNFR JNK signaling through glycosylation.
Thank you for that nice pronunciation of my name. That was pretty correct, actually. So I'm Geert de Vreede. I'm in David Builder's lab, and I'd like to thank the organizers here for giving me the opportunity to talk about my work on the Drosophila tumor suppressor TID Alg3 and how it controls JNK signaling through glycosylation. So one of the questions we want to answer in the Builder lab is trying to see how tumors overgrowth is driven. And basically, I'm very interested in how or what pathways underlie the transformation from a very tightly regulated epithelium and how that transforms towards a more malignant tissue that you see in a tumor where you lose your polarity and you get a lot of overproliferation. And Drosophila is actually a really nice model to study these types of tumors because when you have even just a single mutation in, for instance, a polarity gene, you can get these tumors that form in the imaginal epithelia, as you can see here in the eye disc. But not only that, if we compare it to different human tumor types, you see a lot of similarities, as in you lose the polarity, you see a lot of disorganization, but also the underlying signaling pathways are very similar. So the tumor suppressor gene that I'm interested in, or that I'm very interested in, is the tumorous imaginal discs. And for those of you that know your classic tumor suppressors, this is one that has been described in the early 90s, but not a lot of follow-up research has been done on it. And we actually got really excited about this tumor suppressor when we sequenced some of the alleles and found out that the gene that was attributed to the defects in tumorous imaginal discs was actually miscloned. And we found that there was a different gene responsible for this phenotype. So just to confirm that what this phenotype actually means is when we look at TID tumors, we see that they're slightly, there's the mouse, so slightly overgrown, they lose their structure, but if you look at their cells, you do see that they have more cells than a wild-type wing disc. And when you take these tumors out of the larva just die in early pupal stage, but if you take them out and transplant them into a wild-type adult host, they keep growing and actually kill the host. And as you can see here, they get a lot bigger and they actually have unlimited proliferative potential. As I said before, the gene was miscloned at the time. What we found is that TID alleles actually have mutations in Alg3. And Alg3 is a part of the N-linked glycosylation pathway. So the N-linked glycosylation pathway is a pathway that happens in the ER where a sugar chain gets built up, as you can see here. Different components or different sugar groups get built onto the sugar chain, and Alg3 is just one of the transferases that puts on these sugar groups in this chain, and then it continues to be built up, and in the end, the sugar chain gets transferred to a protein in the ER. Now, just to confirm, we looked in these mutants at glycoproteins. So one of those glycoproteins is K-optin, which has multiple glycosylation sites. And if you look at these proteins that get glycosylated in these mutants, you see increased mobility. So you see a shift on a band, which indicates that you lose this glycosylation in these mutants, and therefore our mutants have impaired glycosylation. So what does N-glycosylation do, or how can a loss cause any tumor? So N-glycosylation mainly affects transmembrane proteins, so that would be a very good potential target to look at. But it can also affect protein folding in the ER. It can affect protein trafficking, interactions and binding of proteins at the membrane. So there's a lot of things that can be affected. So what is actually the main question of my project is to see how does N-linked glycosylation suppress tumors, or how does the loss cause tumors in these Alg3 mutants? And the way we went about this is looking at the different oncogenic pathways that are altered in glycosylation-deficient tumors. So one of the pathways we looked at was the junk signaling pathway. So junk can be activated by different upstream inputs like cell stress or different cytokines and in different developmental contexts. And it activates this MAP kinase cascade that eventually leads to the formation of the AP1 complex that transcribes downstream junk target genes like MMP and Puckert. And when we look at different junk pathway readouts in these tumors, we can actually see that, so for example, a readout like AP1 GFP is very strongly activated in these tumors. And also, if you look more directly at phosphorylation of junk, so the Drosophila junk basket, you stain for phosphorylated junk, and in a wild-type disc, you don't see as much activation, but in Alg3 mutants, you get a lot of it. So junk is clearly activated in these tumors, but is it also functionally relevant? So to answer that question, we took a system where we drove any construct of interest in these tumors with hedgehog GFP. And then, for instance, here we use junk dominant negative to block junk signaling in these tumors. So if you just express this construct, junk dominant negative, in a wild-type disc, you don't affect the disc in any way. But if you do this in one of these Alg3 tumors, you can see that the growth defect or the overgrowth of the tumors is reduced by a lot. But also, if you zoom in on these tumors, you can see that the actual structure reverts more towards wild-type when you're blocking junk just on that side of the tumor. And a thing that I'm not going to go into as much today in the interest of time is that we're actually also seeing that this overgrowth of these tumors is dependent on junk or on hippo signaling downstream of this junk signaling. But the main thing I want to focus on here is to see how junk is ectopically activated by the loss of glycosylation in Alg3 tissue. So then we set about on seeing how do you get ectopic junk activation in Alg3. Well, I told you before that glycosylation happens mostly on transmembrane proteins, and transmembrane proteins that feed into the junk pathway are actually known as, the Drosophila Eiger, which is Drosophila TNF-alpha, and the transmembrane protein Grnd, which is the Drosophila TNFR. And so this is cool. This is something that feeds into the junk pathway. They are transmembrane protein. And really nice for me is that both of them have glycosylation or predicted glycosylation sites. So these are really good candidates for being affected in these mutants. So what we did then, we used the same system where we knock down these different components in the tumors. And when we look at these different knockdowns, so we knock down, for instance, Grnd, the TNF receptor, in the tumor, in the Alg3 tumor, and we see that we also get this really nice rescue in just that side of the tumor. However, if we get rid of the ligand, so TNF, Eiger in the tumor, we don't get rescue, which is a little strange in that you would get ligand-independent activation, which is would be a non-canonical effect. However, it's been known that Eiger can also be produced in distant tissues and act as a secreted ligand, and in that way influence tumors. So we also try to knock down Eiger in different tissues as the hemocytes or the fat body. And when we get rid of Eiger in the fat body, we do see that we can rescue these tumors really nicely. We actually get a really good reversion of these tumors, and they actually look a lot like wild type. So Eiger is involved in generating these tumors, but it acts from a distant tissue. So yeah, as you can see here, Eiger gets produced in the fat body, acts on Grnd locally in the disc. So then we wanted to figure out in what tissue is the glycosylation actually needed. Is it acting on Grnd or Eiger or both of them? So to address that, we made a UAS construct where we just express Alg3. We express it in different tissues and wanted to see where we get any rescue. So and if we do local expression of UAS Alg3, you can see that you, again, get really nice rescue in just that half of the tumor. So you can locally rescue the tumor by locally rescuing glycosylation. However, if you express the rescue construct in fat body, you don't get any rescue. So this basically points to not the glycosylation of Eiger, but just the glycosylation of Grnd per se being able to affect this phenotype. So then when we zoom in on Grnd, we see that it has a glycosylation site specifically in its Eiger binding pocket, which is really nice because that could affect the signaling directly. However, this is still predicted glycosylation site, so we wanted to see if this actually happens in vivo. So when we express a tagged version of Grindelwald in our larva and then basically treat this with PNGase, which basically clips off the sugar groups off of the protein, you see a shift of the protein. So this means that also in vivo, Grindelwald gets glycosylated. And this is really nice because it points us towards a model where the different states of glycosylation actually lead to different signaling outcomes, where the loss of glycosylation in our Alg3 mutants gives ectopic junk activation because it generates a higher affinity. And to test this model, we went to an approach where I took a tagged version of Eiger, so Eiger venus. I overexpressed this in the fat body of the larva in both a wild type and a mutant background, and then wanted to see if there was more binding of Eiger venus to the different tissues. And you would expect that if Alg3 mutants have different affinity, they would have more binding of Eiger. And this is exactly the case. As you can see here, we have a wild-type disc and a Alg3 tumor. And when we look at Eiger venus, we get a lot of binding in a tumor where we get no binding in the wild-type disc. But then we wanted to take it a step further, where we took Grindelwald and mutated the glycosylation site, so actually actively changing the glycosylation. So here, this Grnd N63A is a glycosylation mutant that we generated. And we took that to a culturing approach where we cultured fat body overexpressing this tagged Eiger venus, and put in these discs where we overexpress either the wild type receptor or the receptor that cannot be glycosylated, and then looked at the different binding of Eiger venus. And the cool thing is that we do see a really obvious difference in the binding of Eiger. So if you just overexpress the wild-type Grindelwald receptor, you get some binding of Eiger on the disc. And if you overexpress a mutated version of Grindelwald, a mutated glycosylation site, you dramatically increase the binding. So this really points to a signaling mechanism where the glycosylation of Grnd actively controls the binding of Eiger and controls downstream junk signaling. So then in summary, I hope that I showed you today that loss of glycosylation in Alg3 mutants leads to junk-dependent tumorous overgrowth, that this ectopic junk signaling in these glycosylating mutants is mediated by both Eiger and Grnd signaling, but that it's specifically glycosylation of Grindelwald that acts as a modulating mechanism for Eiger affinity. And what we want to look at now is not only in these particular mutants, but also in a broader sense that in other situations where junk signaling is important through this receptor, that we also see differences in glycosylation and seeing how that regulates junk signaling. With that, I'd like to thank my lab, the Builder Lab, and especially Holly, who was a grad student in the lab that did a lot of the groundwork for this project. I'd like to thank the Leopold Lab for providing a lot of the Grindelwald-related reagents, funding, and you guys for listening. I'll take your questions.
So there are a lot of ALG transferase type genes. Do they all show tumorous phenotypes?
So we do have, so in the pathway, like the next transferase is Alg9, and when we look at Alg9 mutants, we get a very similar phenotype in the discs. So they also have structural differences. They also can be rescued by knocking down Grindelwald. So definitely there's more components to it.
Have you ruled out the possibility that there could be effect on the, say, trafficking of Grindelwald?
Oh, that's definitely an experiment I want to try, because you can argue that Eiger gets trafficked differently, and therefore you see more Eiger accumulation. So we're trying an experiment where we express other tagged proteins and seeing if they get internalized in a different way.
Hi, I'm sorry if I missed this, but I think you referred to the idea of differential glycosylation of Grindelwald. Do you have evidence that it exists in differently glycosylated states in vivo?
And so, yeah, so we have, I mean, I showed you the overexpression data. We did look at the endogenous protein as well. We just don't have that much antibody to look at a lot of, or do like a lot of Westerns on it, but we have some proof that it does show a shift in vivo as well.
Thanks, Geert, for your talk.